Okay, well, it's a real pleasure to welcome Eva Paluch, who's been awarded the Hooke Medal this year as a young investigator. So it's a real pleasure because I was awarded the very first Hooke Medal back in prehistory, you can imagine, before the BSCB was invented. And it's fantastic to be able to pass it on to someone who is really young. So Eva did her undergraduate studies in physics in Lyon, and then she moved to Paris to the Curie Institute to do her PhD in biophysics. And then she completely missed out doing any postdocs and moved straight to the Max Planck Institute of Cell Biology in Dresden, where she started up her own lab in 2006. And she's been awarded many Young Investigator Awards, ERC Starting Grant, and also in 2013, I had the pleasure to interview her for her current position at UCL, where she's already a professor of biophysics, and she's about 20 or something, so this is quite impressive. And in 2015, she was awarded the Philip Leverhulme Prize for contributions to biophysics. So not to beat that, we're going to give her the Hooke Award. And it's a real pleasure, Eva, to welcome you for this award.
Thank you very much, Anne. It's a fantastic pleasure and honor to get this award, especially from Anne, whom I've known for many years, and I find her work to be very inspiring. And I feel it's a great honor to be on the list of people who have received this award, you being one of them as well. So if this starts, let's see. What I'm going to do today is tell you about some recent work we've done in the lab, but before that, I thought I would just in a few slides take you through my kind of background, where I come from and how I ended up having a lab working on cell biophysics, even though I indeed studied physics as an undergrad. And also to kind of acknowledge and thank the people who have been very inspirational and meaningful for me in defining my scientific interest and career. And so in terms of where I come from, indeed, as Anne mentioned, that works. I did study physics, and I studied mathematics at first, and I was really, really interested and fascinated by the beauty of mathematical formulations and the aesthetics of this in a way. And then about in the third year of my undergrad, I decided that I actually wanted to do something a bit more applied, and I actually wanted to switch to biology, but my tutor back then convinced me to start with physics, which is quite a bit more applied than mathematics already. And after that, as pretty much any young physicist, I decided that maybe I actually wanted to study particle physics, because that's what you want to do as a physicist. You want to understand the origin of the universe. And so I did my first summer internship when I was actually 20 at the Observatory of Paris-Meudon. And as actually interesting anecdote, the first paper I was co-author on was about how fast a star would rotate if it was made of a quark-gluon plasma, and what kind of signature it would have for the kind of observations we may have of such a star. So it was very interesting. And the following summer, I actually did what you should do if you want to be a particle physicist, and I went to CERN, where I spent 3 months working on quark-gluon plasma again, which was mostly coding and trying to figure out how a quark-gluon plasma would behave in a big accelerator. And that was kind of a key summer, because that's when I realized that actually I did not want to be a particle physicist. And so I did not want to be that person here. And also kind of a key moment was when I was writing my report for this internship in the library, and I was kind of strolling through the aisle where they had the PhD thesis. And I looked at one randomly, which was about 600 pages. And the title was Calculation of the 17th Decimal of I forgot what cosmological constant. And when I looked at that, I kind of thought, I would love to understand the mysteries of the universe, but I'm not sure I'm cut for that, because I am not able to spend 4 years calculating the 17th decimal of this important constant. And I'm very much admiring people who do this, but I realized that this was not for me. So that's where I started thinking again about what I was initially interested in, which was applying physics to biology. And kind of by a random set of circumstances, I learned from a very good friend of mine from studies that there was a relatively new master's in Paris called Interface Physics Biology, which was meant for people like me, so for physicists, who wanted to switch, so to say, or not really switch, but apply their physical knowledge to biological questions. So it was really meant for people who had no idea about biology after studying 5 years of physics and who wanted to become biophysicists. And so I did that master's, and I had extremely good and fantastic lecturers. Somebody many of you may know in the audience, Frank Julischer, was teaching us like theoretical biophysics. And then we had people like Andrea Dut teaching us cell biology. It was really wonderful. It was a lot of work because practically we did 2 masters in parallel. And after that, thanks to the people we got to meet through this masters, I managed to find a place to do a PhD at the Curie Institute in Paris, under the supervision of these 2 wonderful people, Cécile Sykes, who's a physicist, and Michel Bornins, who's a biologist. And I really worked half-half in these 2 labs, and which were in the 2 kind of departments of the Curie Institute. Cécile was in the physics department, and Michel was in the cell biology department. So I had really the chance of being kind of divided between these 2 disciplines and putting them together in a way. And I also had the chance of being supervised by Mathieu Piel, who some of you might know is now also a PI in Paris, who was a postdoc back then, and who was a very inspiring person. So he really supervised me for the first year and a half or so of my PhD, because he really taught me how important it was to observe things in the microscope and to really try to quantify everything you can. So he kind of said you have to quantify the hell out of everything you're seeing, and then maybe you understand something. And when I worked then already was actually how the actin cytoskeleton generates forces in vitro assays. So these were pushing forces, but also in cells and cellular fragments, and these were contracting forces. And what we were trying to understand really is what kind of forces in general, based on its physical properties, can the actin cytoskeleton generate? And this is really what we're still doing in my lab. And that's just one example of movies we were taking back then, where the actin cortex at the surface of this fragment kind of contracts and make these things that we used to call bulges and that later on we accepted to call blebs, which is something we investigated quite extensively in the lab during the first 5 or 6 years. And so after that, again, by a very random and weird set of circumstances, as Anne mentioned, I actually was lucky enough to skip the postdoc, indeed, and to move directly to this wonderful place, the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden. And I really have to acknowledge here that this really happened thanks to Joe Howard, who was one of the directors there, and who knew me from a couple of conferences, and basically he really talked me into applying for that position in spite of it being relatively early as far as careers go. So I was very lucky to get that place. It was actually a co-appointment with a Polish institute, which has a lot to do with the story because I'm originally from Poland. But anyway, I moved there, and this was an absolutely fantastic place to work on interface questions such as shape of things, because that's what the entire institute was working on, the shape of something. And also because pretty much everybody in this building, which is a biology building, works in very close collaboration with physicists at the neighboring Max Planck, which is headed by Frank Julischer, combining modeling with experiments to understand key questions in cell and developmental biology. And I'm going to give you some examples of that in the rest of the talk. And so finally, after about 7 years there, I moved to the LMCB in London, where we've been actually working together with the Faculty of Physics at UCL in setting up something a little bit similar to the things I've been involved in at Curie and in Dresden. And we're trying to basically enhance collaborations between physics and biology. And we recently set up an Institute for the Physics of Living Systems, which is meant to bridge the gap kind of between physics and biology and bring the 2 together. So why do we do this? Well, why should one take biophysical approaches if we want to study things like cell shape? Well, in fact, the answer is really simple in a way. It's because the morphology of anything and living objects are part of that, is actually inherently and fundamentally a biophysical question. And that's something which was already very clearly acknowledged in this beautiful book, which celebrates its 100 years this year, by Darcy Thompson on growth and form, where he debates the shape and pattern formation in living matter, organism mostly. And he actually spends the first chapter debating whether the laws of physics can and should be applied to understanding shape in biology. And the reason this was a question back then, apparently, based on this first chapter, is that because there was the question of whether it was enough or whether there was something imprinted by God, I suppose, to make these patterns happen. And so what Darcy Thompson concludes after this discussion in his first chapter is that actually the form of any object, be it dead or alive, is in the end a diagram of forces at the surface of this object. And so if we want to understand shape in biology, we need to understand what forces are generated at the surface of the object we're trying to investigate. And so this is true for our bodies, which are to a great extent shaped by bones with bear forces and muscles with exert forces. This is true at the level of tissues, and as I think everybody in this audience knows, there's a number of examples of beautiful morphogenetic processes all through development. For example, here during Drosophila gastrulation, where all these cells here at the ventral side of the embryo kind of contract and form an invagination. And if you would look more closely at the very bottom surface of the embryo, what you see is that this global tissue contraction, which is quite clear in this movie, actually occurs by some small pulsed contraction of the apical surfaces of every individual cell in there. And these contractions are driven by the cytoskeleton, and they really lead to the deformation of this whole tissue. So if we want to understand how tissues form and deform, what we really need to understand fundamentally is how cells themselves control their own shape and how they can affect the different shape changes that they display during morphogenesis in development and disease and all kind of other processes. So this is what we do in the lab. We actually really try to understand the basic principles, not of the universe, but just of cell shape control, which is complicated enough as far as I'm concerned. And we focus on animal cells. So plants have very different principles for shape control because they have a cell wall. But for cells which do not have a cell wall, shape is primarily determined by the cytoskeleton. And for the vast majority of cells out there, not quite all, but almost all, it's really determined by the actin cytoskeleton, because most cells in our organism have underneath their surface, directly attached to the plasma membrane, a very thin and very dense meshwork of actin, which looks like this in electron microscopy. So all these are actin filaments, and it's really deformations and contractions and extensions of this network which drive the vast majority of shape changes we and many of you in the audience are investigating. So how do we understand this? Well, a key problem is that it's a really complicated structure. So it's made of actin, but actually it contains about 150 or a bit more actin-binding proteins. So we have a list now. We've recently done mass spectrometry on that together with the lab of Guillaume Charras. So basically there's a lot of components there. We don't know what all of them do. But for the sake of this talk, there's a number of key components when it comes to the mechanics of cell shape control. And these are the actin filaments themselves, obviously, but also myosin motors, which are non-muscle myosins, so they're a bit smaller than the muscle myosins, and they exert contractile forces on these actin filaments. And finally, cross-linkers, which are quite essential, because if you don't have them, then the whole thing is not connected, and forces cannot be propagated. And so even if we don't quite understand how all these things are arranged and regulated by all these numerous components, what we do know is that if we zoom out and now look at the scale of the entire cell, the whole network has a number of physical properties that can actually be measured. And the most important one for the sake of this talk at least is cortical tension, which is generated in the cortex by myosin motors, which basically constantly try to contract the cell everywhere at its surface. And so what happens is when this tension is locally modulated, for example, if the cell locally accumulates myosin motors to one side of the cell, then there will be more tension there, and then the side will contract. And it's really this kind of processes which drive the vast majority of shape changes that occur in our bodies, such as, for example, cell migration, where actin and myosin accumulate at the back of the cell. We're looking at actin here. And in this case, kind of literally squeeze the cell forward. Or cell division, where actin and myosin accumulate at the cell equator, where they drive the ingression of the cleavage furrow and the separation of the cell into two daughter cells. So ideally, in the grand scheme of things, what we really want to understand is how do we go all the way from here to here, because this is the level that's regulated. So all the signaling pathways that control cell shape or fail to control it act at this level here, because they change either the composition or the activity or the organization of the components of the cortical meshwork. But the outcome we're interested in understanding is what happens at the level of the entire cell. And there's a jump in scales here, which is not very easy to understand from a kind of intuitive level. because these things here happen at the nanoscale. So the whole cortex is a couple of 100 nanometers thick, so it's really not very thick. But the shape changes it drives occur at the scale of the entire cell, which is 10, 20, 30, or even bigger micrometers. So there's like a 2 or 3 orders of magnitude jump here. And so to try to understand this gap scale, what we do is we combine like classical cell and molecular biology approaches with physical measurements whenever possible, because we need to understand how these physical properties are regulated, and so we need to measure them, and also a lot of quantitative microscopy. And we use all this as inputs to physical modeling, which we develop mostly in collaboration with Guillaume Salbreux. I will mention a few times through this talk. And the reason for that is because physical modeling actually provides the ideal kind of tool to bridge this non-intuitive gap, because it really has allows us to understand how putting together a lot of components gives results which are not just the sum of the individual contributions. So what I'm going to do is just give you like a couple of vignettes about a few projects at this scale. So that's kind of from the past of the lab, to give you a little bit more of a background of where we're coming from. And then I will spend the rest of the talk talking about much more recent work on this scale here, which is for the most part at this point not yet published. So in terms of this scale here, so before we started looking at the regulation, we thought we should understand better the mechanics of the cell surface in general. So for example, my two first PhD students, these two guys here, so Mate and Kuba, who actually both recently started their own labs, which is kind of moving. So what they did together is try to understand the mechanics of cell division. And they teamed up for that with Guillaume Salbreux, who's currently a group leader at the Francis Crick Institute, who was formerly a PI in Dresden, but actually at that time when we did that work, he was still a PhD student in Paris. So that was about 12 years ago that this started. But anyway, so the question they were trying to understand, and especially Kuba, who was the first student who joined the lab, was how does this happen mechanically speaking? How does the cell cleave itself into two? So obviously that's a very old question and people have wondered about for many, many decades. But what kind of had not been investigated very much is what most studies had focused on previously was what happens at the cell equator, where forces are exerted to cleave the cell into two. But what we decided to take a look at more closely is what happens at the cell poles, which are vastly ignored generally, because we thought, well, yeah, there are forces exerted at the equator, but actually there is a cortex everywhere as the cell divides. So there's presumably still forces exerted at the poles of the cell. And if these are not perfectly balanced throughout the process of division, then maybe some instabilities can occur where one side would contract at the expense of the other, and that should prevent proper division from happening. And so what Kuba could show is actually this does happen, and there's a number of either genetic manipulations one can make, for example, by depleting a number of actin-binding proteins, or physical perturbations. If one kind of disturbs contractility on one of the poles physically, then basically the poles start squeezing and give rise to this weird oscillation where they kind of each pole empties itself into the other in a periodic fashion, and this usually leads to division failure purely because the forces were not well balanced between the two sides of the cell. And so basically the outcome of that study, which was a very nice combination of like molecular biology with physical perturbation of cells and modeling to understand the mechanics of the system, was that the shape of a dividing cell is actually fundamentally not stable, and that it does have a tendency to do this no matter what, and that actually it needs to be actively stabilized to allow symmetric division to happen in such cells. We also worked quite a lot, and there was about 10 years of studies in the lab on that question, on the mechanics of cell migration, particularly in a three-dimensional context. And so Alba, who was the third student in the lab, who also just started her own lab, worked on this question in collaboration with the group of Carl Philipp Heisenberg, who was back then in Dresden as well, looking at the migration of cells during zebrafish gastrulation. And what she focused on is the mechanics of protrusion formation at the leading edge of here, the precaudal plate, which is the mesendoderm tissue, which basically migrates on the yolk up the embryo. And yeah, we also had a combination of imaging and theoretical modeling to understand how the mechanics of this migration happens. And more recently, Martin, who was also a student in the lab, has actually worked on a similar question, on the question of protrusion formation, but also the mechanics of migration in 3D in cells in culture. in confined environment in culture. And what he could demonstrate, again, using the same tools as usual, so physical measurements and quantitative imaging, is that actually cells which move without focal adhesions, which occur quite a bit if you look at cells moving in vivo, exert way less forces than cells that typically move forming focal adhesions, and that the mechanics of movement in these cells is fundamentally different from a physical perspective from what's known for cells moving on a dish. So these were kind of cell scale studies which we focused on for pretty much for the first 7 or 8 years in the lab. But what I want to talk to you about for the rest of the talk now is how do we go back to molecules from there? So how can we understand how the cell controls at the molecular level, what happens at its surface, or in other words, how do we go from here to here? And for the sake of this talk, I'm going to focus on the cell cycle, where the cell changes shape quite extensively. And what we would like to understand really is how do cells which in interface are usually in a dish spread. If they live in a tissue, they tend to adhere to their neighbors and have all kind of varied shapes. How do they deform and reform and reorganize their cytoskeleton when they enter mitosis to become this stereotypical nice round shape where the spindle can segregate chromosomes? And then how is the cytoskeleton reorganized again to lead to cytokinesis? So we know that this is all driven by changing in actin network organization, because in interface it's typically dominated by this kind of, in this case, stress fibers or cables of contractile actomyosin, which go from adhesion to adhesion, and then all this is disassembled and reassembled into a uniform cortex everywhere around the surface of the cell as cells enter mitosis. So what we would like to know is how this is regulated. So as I told you, there's about 150 actin-binding proteins in these cells. So can we actually understand from the composition what happens? So one first approach could be to understand how this reorganization of actin occurs, is to understand to what extent is the composition of cellular actin network changing when the cell changes shape. So can we get, are there some master switches? Are there some proteins which are not present here and then are present here? And so to answer this question, what Muriel Serre did, who's currently a postdoc in the lab, and this is work which she started when she was in Mark Petronsky's lab before he left to industry. And so basically the question Muriel wanted to ask is, if we look at what binds actin in different phases of the cell cycle, are we going to find some substantial differences, and what are they? So she developed an assay basically when she can isolate using phalloidin-coated beads. So phalloidin is a molecule that binds actin filaments, where she can isolate anything that binds actin filaments from cell extracts from different phases in the cell cycle. And then she did mass spectrometry on that. And what I'm going to do for the next few slides is focus on the transition from interphase to mitosis. She also looked at cytokinesis, but we're still really looking into that. And basically the question is, what proteins bind actin in cell extracts coming from this type of cells, where actin filaments are mostly organized in kind of contractile cables, and what binds actin in extracts that come from mitotic cells, where actin is organized in a uniform cortex around the cell surface. And so she did two separate mass specs on this question, and then she only looked at the intersect and only considered proteins which are enriched at least twofold between in one or the other phase. And so this is the kind of results we get. We basically get a list of proteins which bind either more in G2, so at least 2 times more on the left here, or bind more in mitosis on the right here. And the higher they are on this axis, the more represented they are in mass spec in the extract. So first of all, it's a rather long list. And then, well, it's reassuring that we find some kind of candidates we were expecting. For example, inavas proteins, which are typical regulators of stress fiber formation, are all enriched in interface cells where there is a lot of stress fibers. But then we also find some surprises. For example, all the myosin 2 isoforms are enriched actually in interface and not in mitosis. And yet we know that cells become very, very contractile as they enter mitosis. So there's something we don't quite understand there, and that's something we're currently following up on. Also, something else which is kind of interesting is that we find that intermediate filaments and proteins which bind actin to intermediate filaments are very strongly enriched in mitotic cells. And that's also something which was to some extent surprising, because there were some reports of intermediate filaments being disassembled in mitosis, and that doesn't seem to be the case at all. But also, so that's something we're trying to follow up on to understand what they do. But basically, the summary of all that is that this kind of mass spectrometry analysis gives us big lists of proteins which are enriched that bind actin filaments more in interphase or in mitosis. And what we're doing now is following up on these lists by a number of visual screens, basically, microscopy screens for cell shape and actin organization. But what's clear so we already did a first round of screening in terms of changes in cell shape. And so what's clear is that there is not one switch or not even two switches in there, and that it's much more complex than just increasing the level of one protein or decreasing the level of another one. And so as much as it's really interesting, and I think we're going to be able to understand to some extent how actin is reorganized in this transition taking this kind of approaches, I think what's fundamentally missing and why this will not give us the whole answer is because such lists do not inform us on where these proteins are actually in space and time. And so what we need to do in parallel is also understand what the network looks like actually at the nanoscale and how these proteins are organized with respect to each other in order to understand how the organization of the actin cytoskeleton changes at these cell shape transitions. And so this is what I'm going to spend the rest of the talk on, is trying to understand how the organization of actin networks can be investigated. And because this is a rather broad and complicated question, I'm going to narrow down a little bit and still look at the transition from interphase to mitosis, but focus on cells towards the end of interphase or cells which are detached and rounded in interphase, so that we can compare the interphase cortex, so that we can compare two situations where actin is distributed in the same way, but we know that its physical properties change fundamentally, because it's been shown by a number of labs that actually as cells transition from interphase to mitosis, their contractile tension increases tremendously. And so we go basically from a cortex which is rather weak and not very contractile to a cortex which is very strong and contractile. And what we would like to understand is how this transition in physical properties and possibly actin organization is mediated at the nanoscale level. And so the dogma in understanding this question so far was really that it was all about myosin activity, so that cells become round and contractile as they enter mitosis because myosin 2 is overactivated and possibly expressed more in mitotic cells. But the open question, or something which hasn't been looked at, is to what extent actin itself reorganizes. So this is what we decided to investigate. And the reason it was difficult, it's because, as was already clear, I think, from this confocal picture I showed initially, the actin cortex is a very thin structure. And it makes it, so this is what it looks like in a confocal microscope. And this makes it very difficult to investigate its organization using traditional microscopy techniques. And if you look at it in electron microscopy, it's not very helpful, because this is typically the kind of picture we can get in TEM. This is the bottom side of such a cell here, and this black halo layer here is the actin cortex. So we kind of maybe can measure its thickness from this, but we certainly do not have any information on the organization of anything in there. And also, it's not dynamic. So what we did in the lab instead is try to develop methods to actually measure organization properties and the nanoscale architecture of the actin cortex directly, if possible, in live cells, or at least without using the drastic fixations that are necessary for electron microscopy. So the first thing we did, and this was years ago, before super-resolution microscopes were kind of more widely accessible, is develop a sub-resolution image analysis tool, if you want, to measure the simplest readout of the architecture of the actin cortex, which would be just its thickness. So how thick is this network, and does it change when cells change tension? And so to do that, what we thought is that even if we cannot measure the thickness of a sub-resolution structure using classical confocal microscopy, what we definitely can do is localize it at a nanometer precision by basically labeling the membrane in one color, let's say red, and then actin in another color, let's say green, and then the distance between the two peaks of the green and the red signal should tell us something about the thickness of the cortical meshwork. So actually, it's a little bit more complex than that. One needs to do quite some work to extract cortical thickness from such a measurement, but it can be done. This is the methods describing it. And so basically, we had a tool now to measure the thickness of the cortex just using confocal microscopy. And so what Priyam and Andy, the next generation of PhD students in the lab, did is they asked the simple question I just outlined, which is, if we compare interface cells and mitotic cells, so if we compare cells which have a very weak and low tension cortex to cells which have a very strong and high tension cortex, does thickness change? And does it change in this kind of intuitive maybe way? Is it that interface cells have a thinner cortex and mitotic cells have a thicker cortex? And so we went and measured this, and actually to our surprise we found that it's the contrary. and that mitotic cells have a way thinner cortex than interface cells, and that holds across a number of cell lines we looked at. And this is in spite of the cortical tension, which can be measured by basically squishing them under an AFM, for example, in spite of the cortical tension being quite extensively higher. So that was a bit puzzling, because what we found is basically that the cortical layer is thinner and yet has a much higher tension in mitosis compared to interface. So how could that be? Well, one possibility would be, and that has been suggested in the literature, is that tension is primarily driven by myosin activity, like what we and everybody else thought before, and that actin organization doesn't really matter. But this is not a very satisfying explanation, because in the end, myosins exert forces by pulling on actin. So if actin changes, presumably that does something to the mechanical properties of the cell surface. And so could it be that actually actin network itself, independently of myosin, does something to controlling cell surface tension? So to address this question, what we did is a sort of mini screen, or a targeted screen if you want, on actin binding on a number of actin binding proteins, looking for regulators of cortical thickness that could act independently of myosin. So we first checked if contractility regulators, so myosin itself, they might affect cortex thickness, and we found that at least they do, at least to the significance of what we can measure, they don't. Then we looked at the number of cortex membrane linkers, thinking that maybe if the cortex is more attached to the membrane, it's going to align, and then it's going to get thinner. But depletion of none of these known actin membrane linkers did anything that we could detect to cortical thickness. We also looked at cortex cross-linkers with the same idea that maybe more cross-linking aligns things with respect to the membrane, but it didn't. And then the class of proteins where we finally found some regulators of cortical thickness were mostly regulators of actin polymerization and depolymerization, and thus of actin filament length. And more specifically, what we focused on is the three strongest hits, which were a capping protein, so which caps filaments and prevents their growth. So if we deplete it, it makes filaments longer and the cortex becomes thicker. Severing proteins, which chops filaments into pieces. So if we deplete it, again, longer filaments presumably and a thicker cortex. And finally, formin, which is also an elongator. And if we deplete it, then we get presumably shorter filaments and a thinner cortex. So now we had a number of tools which changed the thickness of the cortical meshwork, and we checked that none of them affected myosin activity. So myosin stays the same. Nothing happens to myosin when we affect these proteins. And yet, when we looked at tension, what we found is that all the depletion of any of these three led to a very strong decrease in cortical tension. So to recap what we had, to recap this in a drawing, is that basically, in mitotic cells, it seems that the thickness of the cortex and the length of the filaments that compose it live at some sort of intermediate optimal length where tension is maximal. And if we make them longer, the filaments, longer and the cortex thicker, or shorter and the cortex thinner, tension drops. So there seems to be a sort of tension optimum for intermediate filament length. So that's a typical example of a result which I find not trivial to interpret just from knowing what these proteins are doing, because if I just look, there's no practically intuitive way of understanding why we would have such a behavior. And so this is a typical case where modeling can help us understand something. And so this is what we did. So Matt, who's a theory postdoc in the lab in collaboration with Guillaume, now in London, developed a computational model of tension generation in a cortex. And in a nutshell, the ingredients of the model are the minimal components we know that are essential for tension generation, which are actin filaments, myosin motors, and cross-linkers. And then if he puts all this together with rules of interaction based as much as possible on experimental measurements, what he sees is that actually we should expect that the surface tension in such a network, which is 3D in the simulation, should behave like this as a function of filament length. It should have an optimum at intermediate filament length, like what we observe experimentally. And the reason for that is that basically when the filaments, the kind of hand-waving reason which we derive from looking at the model, is that when filaments are really short, then things are not connected enough for the network to be able to generate kind of consistent tension. So you have lumps of contracting elements, but nothing is transmitted at the length of the entire cell. And then when filament length increases, tension generation becomes more possible. But then when filaments are too long, what happens is that the whole thing is over cross-linked. And so nothing can be remodeled. The network becomes too rigid, if you want. And so tension is not generated as effectively as it is in the intermediate zones where the network can be remodeled and reorganized by myosins. So the conclusion was that indeed, changing actin filament length keeping everything else, including myosin activity, constant, can change cell surface tension. And that having a thinner cortex in mitosis might actually be because this is more optimal for tension generation in the specific phase of the cell cycle. However, and this is really for the last few slides, we do not mean to say that myosin motors don't matter at all. I mean, they are absolutely necessary to generate tension in the first place. And so from looking at this kind of architectural considerations, we started wondering, how about myosins? How do they actually interact with actin? Because we realized that it's quite crucial to understand how actin and myosin are organized in space to understand how tension is built up. And the reason this became a very puzzling question, which started fascinating us in the lab, is that if I look at the cortex, so this is the electron microscopy picture I showed you before, where you see that it's a very dense meshwork, and it's dense to the point that the mesh size is about 20 to 30 nanometers. And now if I were to draw a myosin mini-filament to scale on that network, this is what it looks like. So that's a myosin, that would be a myosin hexamer, just how it's two heads on each side. And that would be about 300 nanometers long already. And actually, as far as we know, myosin in the cortex is assembled in mini-filaments, which have 15, 16 heads on each side. So you have these structures which are like 350 nanometers long, which somehow generate tension in actin meshwork, which has a mesh size 10 times smaller. So that made us wonder, well, does myosin fit in there? And how far does it go? Can it actually fit spatially in this meshwork? And so this is what Binyan, who's a engineer postdoc in the lab working on super resolution microscopy, took up as a project. And so what he did, now that we had moved to London and had access to a wonderful super resolution microscopy facility, is he started looking at how actin and myosin localize with respect to each other using dual color storm microscopy, which has like a 50 nanometers resolution in the kind of situation we're looking at. And so when he looks at mitotic cells, what he saw is that actin and myosin indeed nicely overlap. But when he looked at interface cells, which have a thicker cortex, he could see that actually they don't overlap, and that there seems to be, so myosin is in purple or magenta, that there's a whole region of the cortex which is green without myosins in there, and that there seems to be a sort of overhang of myosin in the cytoplasm, which we hypothesize is myosin mini-filaments which are bound with one side, and the other side is kind of hanging in the cytoplasm and not bound to the cortex. And interestingly, he can modulate to what extent the extent of this overlap by changing myosin activity. And if he makes myosin overall less active by expressing a non-active mutant, he can see that this overhang becomes even higher, so myosin just doesn't seem to be getting into the cortex at all, practically, in these cells. And if he makes myosin more active, then the overhang almost disappears. And so basically, and this can be all quantified, basically what this means is that myosin penetrates more or less into the cortex depending on its activity. And that as far as, so that's something we're pursuing right now, but as far as we understand so far, it seems to be purely because of steric effects due to the size of myosin mini-filaments and the density of the cortical meshwork. And finally, what we started doing recently, and that's just kind of a snapshot, is that actually what we realized is that myosin being as big as it is, we might actually be able to see it just directly in the cortex. And so to do this, we used a beautiful construct which was given to us by John Hammer from NIH, where myosin heads are labeled in one color and myosin tails are labeled in another color. And then if you overexpress this in cells, one should be able to see mini-filaments, which would be structures with a green, magenta green kind of blob succession at the right kind of distance. And so if we express this in mitotic cells in this case and squish them a little bit so that we have a nice surface of cortex to look at, what we can see is actually individual myosin mini-filaments. This can be done live because this is done using structured illumination microscopy, basically moving around and exerting contractile forces in the cortex. So now we can really actually quantify what individual myosins are doing and how they move or not move to generate tension at the surface of the cell. So the conclusion of all this is that cell surface tension, which for a long time, including by us, was thought to be mostly regulated by the level of myosin activity, by the kind of stresses myosins can generate, is actually regulated at a multitude of levels, and that we have to take them all into consideration if we want to understand how cells contract to change shape. Because in addition to stresses generated, myosin activity is also going to control to what extent myosin itself penetrates into the actin cortex. And if there's too big of an overhang, then all these myosins, which are half hanging in the cytoplasm, are not going to be contributing to tension generation in these cells at all. We've also shown that the Organization of the actin meshwork itself is actually quite essential, and if we change that by changing, for example, filament length or network connectivity, then tension is going to change independently of myosin. And then there's other levels we're looking at currently, such as the alignment of myosin mini-filaments with respect to the plasma membrane, which will also modulate how much tension is generated depending on how they are oriented in the cortex. And so what we hope to do in the future is kind of really put all this together. So these super resolution kind of approaches where we can localize different cortex components with respect to each other, with electron microscopy approaches where we hope to finally find ways to look at actin filaments themselves, which is still the bottleneck. We cannot visualize them at this point using any technique that I know of. Using some super resolution live techniques where you can, so these are myosin mini-filaments again. In this case, they are moving in the cortex. And use all this now as proper inputs into this kind of computational models of the cortex where we can now actually have actual data on the dynamics of myosin mini-filaments, for instance, and also cross-linkers and hopefully actin itself to really understand what happens when cells go from being round to being, for example, dividing. And I'm going to stop here and thank again everybody involved. So I showed you the pictures of some key people in the lab. I haven't covered, obviously, all the projects we've worked on. So I'm extremely thankful to my lab present. The green people are the people from the projects I described, and past over the last 13 years almost, who have been all tremendous and amazing. And without them, none of this would have happened. They've always been a wonderful combination of physicists and biologists and engineers working together, and it's a lot of fun. I'm also very thankful to a lot of collaborators, particularly Guillaume Salbreux, who's the theorist I mentioned a few times, with whom we've been working consistently for the last 12 years. And it's been an amazing collaboration. Philippe Roux and Mark Petronsky were both involved in the mass spec studies I present, and as well as Matthias Samwer, who developed the beads for the pull-down assays. And finally, we rely very heavily, we did in Dresden and even more so in London, on imaging facilities, particularly the super resolution facility and the electron microscopy facility at the NMCB. And I would like to thank you for your attention and thank the BSCB again for this wonderful medal.
Thank you very much, Eva, for that fantastic talk. Are there questions? Yeah.
What happens if you give cytoplasm from cells in specific cell cycle stage to pure actin?
So what do you mean? If you take...
If you were to take cytoplasm from cells in a specific cell cycle stage to pure actin or actin with something that looks like membranes,
right.
do you get that type of...
Do you get different types of networks? You mean?
Yes.
Yeah. So surprisingly, that, as far as I know, hasn't been done. So that's kind of what we do with the beads, and we're currently trying to do electron microscopy on these, so I don't know the answer. But we're trying to do exactly this using scanning electron microscopy on these extracts to see if there's any difference in network organization. There definitely is a difference in network composition. So what that's going to do to organization, I don't know.
Okay, thanks.
So Eva, I was very interested in this thing when you said the active, when you make myosin more active, you lose it or it gets tighter with the actin. Is that because it's binding to the actin more tightly, or is it turning over ATP faster, or is it a mixture of both? What do you mean by more active?
Yeah, right. Okay, so there's different ways we try to do more active. So there's, the one I was showing you is a phosphomimic mutant of myosin regulatory light chain. So in a mini-filament, presumably, there are some of the myosin heads which are active, so being able to bind actin, and some which are not. But that's not that well understood, actually, what happens with these. So in vitro, it's been studied, but in cells, we don't really know. And so what I think, because myosin 2 is not processive, right? So the only reason it can actually exert forces in a consistent way is because it forms mini-filaments. And I guess the number of heads which are active at a given time modulates to what extent the process. So I suppose it's by processivity changes, effective processivity changes. And the fact that it gets tighter, I think, but I have no evidence of that. But the only way I can understand this, and I'm actually happy if anybody has any other idea, is that the reason there's an overhang is because you probably have many filaments where one side is bound and the other isn't. And if it's more active, then there's more chances than this other side becomes bound when it's in proximity to the cortex. Does that make sense?
So with the very beautiful pictures of the cells dividing symmetrically, I was starting to wonder what the forces are when they divide asymmetrically and how it has to change.
Yeah, that's a great question. So there are some studies in neuroblasts suggesting that actomyosin, maybe even just contraction at one pole, can lead to asymmetries in shape. I think so. That's something we're actually pursuing in the lab right now, is the question of how symmetric is symmetric, because there's no such a thing as perfectly balanced forces. So presumably asymmetric division has some level of variability in the sizes of the two cells just because of the unbalanced contractility of the two poles. And there might be situations where this imbalance is made larger, such as maybe in neuroblasts, where this would lead to controlled asymmetries. Now, what's completely unclear to me is how do you control such an asymmetry? Because once it starts contracting, from a purely physical perspective, it should just collapse. So there must be some regulatory processes, possibly from the spindle, which kind of set a different shape. Because obviously the spindle signals to all that, so it's quite key in maintaining the symmetry as well.
Okay, thank you very much again to Eva Paluch.